So in this example of glycogen phosphorylase, folks, we are going to see covalent modification as a way to allosterically control an enzyme. In addition to allosteric control, we will also see a little bit of control at the active site as well, and I'll do my best to mention that or allude to it as we see it. But before we talk about covalent modification, what type of covalent modification are we talking about in this example? It's phosphorylation. This is an extremely common example of covalent modification of proteins. About 30% of all proteins in our bodies can be phosphorylated or dephosphorylated. It's going to happen at the hydroxyl of either serine, threonine, or tyrosine. The proteins that perform the phosphorylation are called protein kinases. Proteins that remove the phosphate or dephosphorylate are called phosphatases. And what generally is going to happen, folks, is we'll take that R group and throw a phosphate on it. The protein kinase uses ATP, converts it to ADP to stick that phosphate on, and the protein phosphatase will generally hydrolyze off that phosphate with water, getting us back to the dephosphorylated version. And just to make this really clear, because it's kind of a picture on this slide, let's draw out what this might look like for serine, for example. So here's our good friend serine dephosphorylated. Now if we were to phosphorylate it, it would look like this. So we have an idea what we're seeing in our mind's eye when we're discussing phosphorylation of a protein. Literally, we just pop a phosphate onto the hydroxyl group. In this example was serine. So let's jump into the details here, folks. The enzyme we're discussing now is called glycogen phosphorylase. Its nickname is phosphorylase, so just phosphorylase. This enzyme is important for all of us during times we are not actively eating. It catalyzes the breakdown of sugar storage in our body, so glycogen, to glucose 1-phosphate. So substrates are glycogen and phosphate, and the product is a shorter glycogen molecule and glucose 1-phosphate. Let's keep in mind that glycogen is basically polymerized glucose. It's the way that our body stores glucose. So how is this enzyme controlled? It's a little bit complicated, folks, so we'll walk through step by step and then get a big picture at the end. So here is phosphorylase. Phosphorylase is inactive, but to turn it on, we can have high levels of AMP. AMP is a low energy signal. Why does it make sense that a low energy signal would turn this enzyme on? Well, the whole point of this enzyme is to get us energy, right? It's breaking down glycogen in our body to provide us with a source of glucose. So, if AMP is a low energy signal, turns phosphorylase on, to provide us with glucose. That makes sense. On the other hand, both ATP and glucose 6-phosphate, G6P, these are signals that there is energy around. And if we already have enough energy, we don't need more. So we can turn the enzyme off. And these work through feedback inhibition. Now, in comes the phosphorylation of the enzyme. So what happens to the regulation of this enzyme when it is phosphorylated? So when the enzyme is phosphorylated, it tends to be always on. So phosphorylating this enzyme is a signal to turn it on. We need energy. So a phosphorylation event signals more energy is needed. Get this enzyme turned on and breaking down that glycogen, releasing glucose. However, if the enzyme is phosphorylated, now glucose becomes an inhibitor. Why does this make sense? Well, if we already have a lot of glucose around, we don't need to break down glycogen to make more glucose. And so here's the slide with kind of everything put all together. Okay, folks, we have phosphorylase. Please do not worry about phosphorylase A, phosphorylase B. We're just talking about glycogen phosphorylase. When phosphorylase is phosphorylated by the kinase, it tends to be on. However, if there's a lot of glucose present when it's phosphorylated, it'll turn back off. When we dephosphorylate the enzyme, it's more likely to be off. But if we have AMP, that tells us we're in a low energy state. We got to turn back on, get some glycogen converted to glucose. But if we have high energy signals, if there's lots of ATP around or lots of glucose 6-phosphate, we're going to turn this enzyme back off. We don't need more energy. Now, folks, to wrap up this kind of higher level discussion of this enzyme, I want us to think about competitive inhibition. And for a moment, I'm going to take us back to this slide here. So the substrates of this enzyme are glycogen, which is a polymer of glucose, and phosphate. And the product of the enzyme is glucose 1-phosphate. The structure is given to you on the screen. I want you to look at some of the small molecules discussed on this slide: ATP, glucose 6-phosphate, AMP, and glucose, and think to yourself, is it possible that any of these types of inhibitors could be competitive in nature? Something to ponder. To wrap up this case study, folks, I want to link some structural aspects of the enzyme glycogen phosphorylase to its inhibition or activation. And so here on this screen, on the left is a structure of the T state, on the right is a structure of the R state. Here you're seeing the tertiary structure, mostly alpha helices, some beta sheets for sure, and of course turns and loops. There are two locations I want us to focus on. Kind of up here at the top is the allosteric site, and then here in the bottom, kind of within the protein more, that's the active site, okay? What happens if the enzyme is phosphorylated at serine 16, or if the enzyme has bound to AMP? What happens when this happens? What happens when AMP is bound or when serine is phosphorylated is that the N-terminus that was down here swings up and reorients itself up here. This is because there is an arginine residue that can bind to that phosphorylated serine. Recall, the phosphate has a negative charge. Arginine is positively charged. So something as simple as a single phosphorylation event will cause the tertiary structure to change in such a way that the N-terminus completely shifts where it's located. But as we know from hemoglobin, when one part of the tertiary structure changes, other parts of the tertiary structure changes too. So when the allosteric site changes shape, the active site changes shape. So you can see a difference between where the arginine and the active site is located, okay? This is a separate arginine than the one in the allosteric site. So the arginine in the active site also changes where it's located. Well, this makes binding of the phosphate substrate better. So here we're talking about making the Km stronger, making the Km smaller. So changes at the allosteric site impact the structure of the active site, making binding of substrate better. And other shape changes happen too, right? Let's not worry too much about this, but there's a little bit of a green loop that I'm highlighting here that's in a different place in the R state versus T state. There's an alpha helix highlighted here in blue that's in a different place, T state versus R state. All to just remind you folks that structural changes in one spot in an enzyme will impact the entire structure of the enzyme. Something as simple as a phosphorylation of a single residue in a protein can cause these changes to happen. And that wraps up our time together, folks. Kinases are incredibly important for signaling. If you take Cell-Molec, you've probably learned a kinase cascade because so much signaling within cells is done via adding a phosphate or removing a phosphate. Not to make it even more complicated, but the kinases that perform phosphorylation and the phosphatases that remove phosphates are themselves often regulated by phosphorylation from other kinases and phosphatases. That's all for allosteric modulation and inhibition for today. We talked about allosteric inhibition of an enzyme called ATCase. We talked about why it makes sense that ATCase is inhibited or activated. We also saw for glycogen phosphorylase allosteric inhibition through allosteric activation through phosphorylation, but we saw inhibition and activation through a variety of small molecules as well. And hopefully we learned more about structure function for CTP, where its quaternary structure makes it more cooperative when inhibited, and for glycogen phosphorylase, where we saw tertiary structure changes leading to its activation. And I'll leave you with today's cat meme. See you next time.